Hey everyone, and welcome to today's Explainer. We're jumping straight into a highly structured, deep dive into the biochemistry of lipids. So if you're gearing up for A-levels or board exams, or honestly just want to master the molecular foundations of life, you are absolutely in the right place. We're taking this strictly from textbook theory right down to the laboratory bench. Exactly. We're not just going to sit here and memorize facts today. We're going to see exactly how these molecules actually behave in the real world, and more importantly, in your lab experiments.
Right, but before we get bogged down in all the heavy chemistry, let me hit you with a quick real-world hook. Take the desert jerboa. This tiny little guy survives in this incredibly harsh, dry environment without ever actually drinking water. No way, right? Well, how does it do it? It relies almost entirely on something we call metabolic water. Basically, when the jerboa breaks down its stored triglyceride lipids through cellular respiration to make ATP, water is literally created as a direct chemical byproduct. It's essentially manufacturing its own water supply entirely from its body fat. How cool is that?
Okay, so here's our roadmap for today's explainer. We'll start with the architecture of lipids, move on to saturated versus unsaturated fats, and then look at forming the ester bond. After that, we dive into phospholipids and cell membranes, tackle the lipid emulsion test, and finish up with a 10-second exam summary. Let's get right to it.
Section 1. The architecture of lipids.
Let's kick things off with a core definition of a triglyceride. Now, this is the most common kind of lipid you'll encounter, usually functioning as a massive energy storage molecule. Simply put, a triglyceride is a macromolecule composed of one glycerol backbone that's covalently bonded to 3 fatty acid chains. You can kind of think of it like a coat rack. The glycerol molecule, which is an alcohol, is the sturdy wooden rack, and those 3 fatty acid hydrocarbon chains, they're the coats hanging off of it.
And right away, we need to hit our first high-yield corner warning. Listen up, because this is a massive exam trap. Lipids are macromolecules, yes, absolutely, but they are not polymers. Unlike carbohydrates or proteins, which are literally built by chaining together identical monomers over and over again, lipids just don't have that repeating chain. They're built by joining different simple structures, like that one glycerol and 3 distinct fatty acids, into one complex molecule. So whatever you do, do not call a lipid a polymer on your exam.
Section 2. Saturated versus unsaturated fats.
Okay, so how does that molecular structure actually dictate physical state? Like, why is the fat on a piece of steak completely solid, but the olive oil sitting in your kitchen is liquid? It basically all comes down to the carbon bonds inside those fatty acid chains. Saturated fatty acids only have single bonds between their carbon atoms. Because of that, they hold the maximum possible number of hydrogen atoms. They are literally saturated with them, and this makes the hydrocarbon chain perfectly straight. Unsaturated fats, on the other hand, contain at least one double bond between carbons. That double bond means fewer hydrogens, sure, but more importantly, it physically causes the chain to bend, or kink. Right, and the crucial point here is how that geometry plays out physically in the real world. Because saturated fats are completely straight, those triglyceride molecules can pack incredibly close together. This allows for much stronger intermolecular bonding, specifically van der Waals forces, which gives them a higher melting point. That's exactly why they're solid at room temp. Unsaturated fats, with those bent, kinked chains, they just can't pack tightly together. The bonding between them is much weaker, so they stay loose and fluid. Boom! Liquid oils.
Section 3. Forming the ester bond.
So we know the components now, but how do we actually glue them together from scratch? Well, we link them up using a condensation reaction. Here's how it happens. Step 1. The hydroxyl group, the OH, on the glycerol molecule comes into close proximity with the carboxyl group, the COOH, of the fatty acid. Step 2. They react, and a molecule of water, H2O, is chemically removed. Step 3. Because they're both missing a bond now, the oxygen and carbon link up to hold hands, forming a really strong covalent bond known specifically as an ester bond. I'm going to jump in here and drop another high-yield trap right on the lab bench. Notice the math here, guys. Because there are 3 separate fatty acids on a single triglyceride molecule, you have to do this condensation reaction 3 times. That means exactly 3 water molecules are released for every single triglyceride synthesized. So if an exam question asks you how much water is produced when making 10 triglycerides, your answer had better be 30.
Section 4. Phospholipids and cell membranes.
All right, let's shift our focus away from energy storage lipids like our triglycerides and look at structural lipids, specifically the architects of the plasma membrane. Now, a phospholipid is incredibly similar to a triglyceride, but instead of 3 fatty acids, it only has 2. That third position on the glycerol backbone is taken up by a polar phosphate head.
And guys, this is where we introduce a massive board exam buzzword, amphipathic. Because that phosphate head has a charge, it's polar and hydrophilic, meaning it loves water. But those two fatty acid tails, they remain nonpolar and hydrophobic, so they absolutely hate water. When a molecule possesses both of these contradictory properties at the exact same time, it is amphipathic. In the watery environment of our bodies, these molecules spontaneously arrange themselves into a bilayer, basically hiding their tails on the inside and pointing their polar heads outward to interact with the water.
Now, let's actually apply this in a functional lab context, membrane fluidity. Remember that straight versus kinked chains concept we talked about? If your plasma membrane is composed of phospholipids with lots of saturated, straight tails, they pack tightly, creating a very rigid, solid membrane. But if you introduce more unsaturated fatty acids with double bonds, those bent legs prevent that tight packing. And this loose arrangement is absolutely essential. It ensures the cell membrane remains fluid, flexible, and essentially doesn't freeze solid the second temperatures drop.
Section 5. The lipid emulsion tests.
Speaking of lab, how do we actually prove that nonpolar lipids are present in some mystery sample? Well, we run the lipid emulsion test. And here is your exact benchwork protocol. Step 1. You take your sample and add pure 95% ethanol. Step 2. Shake vigorously. You see, ethanol is completely unique. It can actually dissolve nonpolar lipids. So if lipids are in there, they dissolve right into the ethanol. Step 3. You pour this new mixture into a completely separate test tube filled with plain water. Step 4. Just observe. Because lipids are extremely hydrophobic, the literal second they hit the water, they desperately try to escape it. They clump together into these tiny droplets, physically separating from the water. These droplets then scatter light in all directions, creating this highly visible, cloudy white emulsion.
Now, if you get a data analysis question on your exam about this, you really need to know your experimental design. Your independent variable is the actual substance you're testing, say vegetable oil versus some unknown solution. Your control group is incredibly vital here. You run the exact same test, but you replace the lipid sample with pure water, just to prove that the ethanol and water alone don't turn cloudy. Finally, the data analysis. A cloudy white emulsion is a definitive positive result for lipids. If the solution remains totally transparent, that is a negative result.
Section 6. The 10-second exam summary.
All right, we've made it to the end. It is time for the summary. We're going to rapid-fire the absolute must-know, highest-yield bullet points. Number 1. A triglyceride is made of 1 glycerol and 3 fatty acids joined together by ester bonds.
Number 2. Saturated fats are perfectly straight and solid at room temp. Unsaturated fats have double bonds, causing them to kink, which makes them liquid.
Number 3. Phospholipids form cell membranes because they're amphipathic, featuring a polar, water-loving head and nonpolar, water-fearing tails.
And finally, number 4. To test for lipids, run the emulsion test. Add ethanol, then water, and look for that positive, cloudy white emulsion.
Real quick, before we wrap up this explainer, I want to leave you all with one final, kind of provocative thought. We learned earlier that unsaturated fats are generally liquids, right? And they're considered healthier. But what about trans fats? Structurally speaking, they have double bonds, so they are technically unsaturated. Yet, because the hydrogen atoms are on opposite sides of the double bond, the chain somehow remains bizarrely straight. And because of this completely unnatural shape, they can't properly form enzyme-substrate complexes in our bodies to be metabolized. This leads them to pack tightly and build up as plaque in our bloodstream, heavily linking them to coronary heart disease. It's just a perfect example of how one tiny molecular shift can entirely rewrite biological function. Keep questioning the chemistry, everyone. Thanks so much for hanging out with us, and we'll see you in the next explainer.